Lesson 9, Lipids. Lipids include a diverse group of compounds that are largely nonpolar in nature. This is because they are hydrocarbons that include mostly nonpolar carbon-carbon or carbon-hydrogen bonds. Nonpolar molecules are hydrophobic or insoluble in water. Lipids perform many different functions in a cell. Cells store energy for long-term use in the form of fats. Lipids also provide insulation from the environment for plants and animals. Lipids are also the building blocks of many hormones and are an important constituent of all cellular membranes. Lipids include fats, oils, waxes, phospholipids, and steroids. Here's some more chemistry for you. A fat molecule consists of two main components, glycerol and fatty acids. Glycerol is an organic compound with 3 carbons, 5 hydrogens, and 3 hydroxyl groups. Fatty acids have a long chain of hydrocarbons to which a carboxyl group is attached, hence the name fatty acid. The number of carbons in the fatty acid may range from 4 to 36. The most common are those containing 12 to 18 carbons. In a fat molecule, the fatty acids attach to each of the glycerol molecule's 3 carbons with an ester bond through an oxygen atom. We also call fats triacylglycerols or triglycerides because of their chemical structure. Fatty acids may be saturated or unsaturated. In a fatty acid chain, if there are only single bonds between neighboring carbons in the hydrocarbon chain, the fatty acid is saturated. Saturated fatty acids are saturated with hydrogen. In other words, the number of hydrogen atoms attached to the carbon skeleton is maximized. Stearic acid is an example of a saturated fatty acid. When the hydrocarbon chain contains a double bond, the fatty acid is unsaturated. Oleic acid is an example of an unsaturated fatty acid. Most unsaturated fats are liquid at room temperature. We call these oils. If there is one double bond in the molecule, then it is a monounsaturated fat, like olive oil. And if there is more than one double bond, then it is a polyunsaturated fat, like canola oil. When a fatty acid has no double bonds, it is a saturated fatty acid, because it is not possible to add more hydrogen to the chain's carbon atoms. A fat may contain similar or different fatty acids attached to glycerol. Long, straight fatty acids with single bonds generally pack tightly and are solid at room temperature. Animal fats with stearic acid and palmitic acid, common in meat, and the fat with butyric acid, common in butter, are examples of saturated fats. Mammals store fats in specialized cells, or adipocytes, where fat globules occupy most of the cell's volume. Plants store fat or oil in many seeds and use them as a source of energy during seedling development. Unsaturated fats or oils are usually of plant origin and contain cis-unsaturated fatty acids. Cis and trans indicate the configuration of the molecule around the double bond. If hydrogens are present in the same plane, it is a cis fat. If the hydrogen atoms are on 2 different planes, it is a trans fat. The cis double bond causes a bend or a kink that prevents the fatty acids from packing tightly, keeping them liquid at room temperature. Olive oil, corn oil, canola oil, and cod liver oil are examples of unsaturated fats. Unsaturated fats help to lower blood cholesterol levels, whereas saturated fats might contribute to plaque formation in the arteries. The food industry artificially hydrogenates oils to make them semi-solid and of a consistency desirable for many processed food products. Simply speaking, hydrogen gas is bubbled through oils to solidify them. During this hydrogenation process, double bonds of the cis conformation in the hydrocarbon chain may convert to double bonds in the trans conformation. Margarine, some types of peanut butter, and shortening are examples of artificially hydrogenated trans fats. Recent studies have shown that an increase in trans fats in the human diet may lead to higher levels of low-density lipoproteins, LDL, or bad cholesterol, which in turn may lead to plaque deposition in the arteries, resulting in heart disease. Many fast food restaurants have recently banned using trans fats, and food labels are required to display the trans fat content. Essential fatty acids are those that the human body requires but does not synthesize. Consequently, they have to be supplemented through ingestion via the diet. Omega-3 fatty acids fall into this category and are one of only two known for humans. The other is omega-6 fatty acid. These are polyunsaturated fatty acids and are omega-3 because a double bond connects the third carbon from the hydrocarbon chain's end to its neighboring carbon. Like carbohydrates, fats have received considerable bad publicity. It is true that eating an excess of fried foods and other fatty foods leads to weight gain. However, fats do have important functions. Many vitamins are fat-soluble, and fats serve as a long-term storage form of fatty acids, a source of energy. They also provide insulation for the body. Therefore, we should consume healthy fats in moderate amounts on a regular basis. Phospholipids are major plasma membrane constituents that comprise cells' outermost layer. Like fats, they are comprised of fatty acid chains attached to a glycerol or sphingosine backbone. However, instead of 3 fatty acids attached as in triglycerides, there are 2 fatty acids forming diacylglycerol, and a modified phosphate group occupies the glycerol backbone's third carbon. A phosphate group alone attached to a diacylglycerol does not qualify as a phospholipid. It is phosphatide, diacylglycerol 3-phosphate, the precursor of phospholipids. An alcohol modifies the phosphate group. Phosphatidylcholine and phosphatidylserine are 2 important phospholipids that are in plasma membranes. A phospholipid is an amphipathic molecule, meaning it has a hydrophobic and a hydrophilic part. The fatty acid chains are hydrophobic and cannot interact with water, whereas the phosphate-containing group is hydrophilic and interacts with water. The head is the hydrophilic part, and the tail contains the hydrophobic fatty acids. In a membrane, a bilayer of phospholipids forms the structure's matrix. Phospholipids' fatty acid tails face inside, away from water, whereas the phosphate group faces the outside aqueous side. Phospholipids are responsible for the plasma membrane's dynamic nature. If a drop of phospholipids is placed in water, it spontaneously forms a structure that scientists call a micelle, where the hydrophilic phosphate heads face the outside and the fatty acids face the structure's interior. Unlike the phospholipids and fats that we discussed earlier, steroids have a fused ring structure. Although they do not resemble the other lipids, scientists group them with them because they are also hydrophobic and insoluble in water. All steroids have 4 linked carbon rings, and several of them, like cholesterol, have a short tail. To conclude this lesson, let's talk about the most common steroid, cholesterol. The liver synthesizes cholesterol and is the precursor to many steroid hormones, such as testosterone and estradiol, which gonads and endocrine glands secrete. It is also the precursor to vitamin D. Cholesterol is also the precursor of bile salts, which help emulsify fats and their subsequent absorption by cells. Although lay people often speak negatively about cholesterol, it is necessary for the body's proper functioning. Sterols, cholesterol in animal cells, phytosterol in plants, are components of the plasma membrane of cells and are found within the phospholipid bilayer. See you in the next lesson.